This is the Chapter 11 video. We're going to talk about antibiotics and resistance. Antimicrobial drugs interfere with the growth of microbes within a host. There's different types of antimicrobial drugs, such as antiprotozoals, antifungals, antivirals. However, we're going to be focused on antibiotics, which target bacteria. These antibiotics need to be selectively toxic, meaning they're going to kill or harm the microbe but not damage the host yourself. Antibiotics were basically discovered by accident. Most antibiotics would be produced by either bacteria or actual fungus. And here we have an environmental sample with basically a lawn of bacteria created from, say, a soil sample plated on a petri dish. And here, where you see these clearing areas around these fungal colonies, that means the actual fungus is producing an antibiotic, secreting it, and killing the microbe. The bacteria around it. Um, these clearing areas are called a zone of inhibition, which we will see in our Chapter 11 lab. Antibiotics have different spectrums of activity. They can either be broad spectrum or narrow spectrum. So broad spectrum target a wide range of microbes, gram-positive and gram-negatives, for example, where narrow spectrum just target a relatively small range of microbes. The use of broad-spectrum antimicrobial drugs also kill normal microbiota. So survivors of your normal microbiota that are resistant naturally to the antibiotic or through mutation, those survivors may become opportunistic. So it can lead to a secondary infection, often referred to as a superinfection. So a yeast infection or antibiotic-associated diarrhea from Clostridium difficile would be some examples of that. So that term superinfection is applied to the growth of a target pathogen that has developed resistance, or a strain that replaces the original strain and the infection continues. Antibiotics have basically 5 different modes of action. So you will want to be familiar with the modes of action of the antibiotic. So the mode of action is how or what the actual antibiotic is targeting on the bacterial cell. So we can inhibit the cell wall synthesis, inhibit protein synthesis, inhibit nucleic acid synthesis, injure the plasma membrane, or inhibit a metabolic process. Inhibiting cell wall synthesis, we're going to weaken the cell wall, and the cell is going to actually eventually lyse. So penicillin, cephalosporins, vancomycin would be some examples of antibiotics that target cell wall synthesis. Here's an image of 2 rod-shaped bacteria before the introduction of penicillin. The bacteria is introduced to penicillin, and you can see that weakening of the cell wall and lysis of that cell because that cell wall is broken. Inhibitors of protein synthesis target prokaryotic 70S ribosomes. So either affecting the actual ribosome or blocking tRNA, things like that. Now, if you remember, mitochondria also have 70S ribosomes. So sometimes the side effect of these protein synthesis drugs would be fatigue because they're actually targeting your mitochondria as well. Some inhibitors of protein synthesis would be your aminoglycosides, such as streptomycin. So that's going to change the shape of the 30S subunit of the ribosome. Tetracyclines, a broad spectrum, which interfere with tRNA attachment. Inhibitors of nucleic acid synthesis. Basically, what's going to happen is you're going to block either DNA replication or mRNA transcription. So usually these drugs would not necessarily be selectively toxic and actually target eukaryotic cells, such as your own cells as well. So rifamycin would be an example of that, inhibiting RNA synthesis. Injury to the plasma membrane or cell membrane. Basically, we're going to have channels form through the cell membrane and damage the integrity. So polymyxin B, which is most people are familiar with because it's combined with bacitracin and neomycin in your neosporins, or triple antibiotic. So it's a topical antibiotic. Antimetabolics, these are going to interfere with a metabolic process of the bacteria. And these can only really be effective when the metabolic process of the pathogen is different from the host. So an example would be your sulfonamide drugs, which inhibit folic acid synthesis. So the bacteria can actually produce folic acid for itself, um, and folic acid is needed for DNA and RNA synthesis. So if it can't reproduce its DNA and RNA, then it's not going to be able to grow. This is selectively toxic because we do not produce our own folic acid. We absorb that through the diet that we consume. Sulfonamide is a competitive inhibitor, meaning it's going to have a similar chemical structure to the substrate that would bind to the enzyme normally. So sulfonamide here has this chemical structure, and that mimics this PABA molecule that will eventually be turned into folic acid in the bacterial cell. You can see a very similar structure. This side chain here is what would differ. So some clinical consideration in prescribing antimicrobials would be some safety and side effects. Toxicity would be one of the main concerns, making sure that the person has the proper dosage because a little bit more toxic than others. So they may be toxic to the kidneys, liver, nerves, and consideration needs to be taken when prescribing drugs to pregnant women because some antibiotics can be toxic to the fetus or they can lead to basically lifelong defects. Allergies would be another concern. They're rare, but they can be life-threatening depending on the severity of the allergic reaction. Here's a couple examples. This is a black hairy tongue caused by antiprotozoal drug Flagyl. This is a temporary harmless side effect. And this staining of the enamel of the teeth, this would be permanent lifelong damage due to a mother taking tetracycline during the fetus development. Now the more commonly interesting topic when we talk about antibiotics is the resistance that some bacteria develop towards them. So some pathogens are naturally resistant. So gram negatives are naturally resistant to certain penicillins because the outer membrane protects the actual cell wall. So resistance is acquired in two ways, either through mutation of chromosomal genes or the acquisition of plasmids through transformation, conjugation, or transduction. We contribute to the problem, the rise of antibiotics, because when we are given an antibiotic, we put selective pressure on bacteria. So we are killing the weak and the strong survive, basically. So if we use outdated or weakened antibiotics, we can contribute to the rise of antibiotic-resistant bacteria. Outdated, some antibiotics actually become more toxic if they're outdated, or they're weakened because they are expired. When we have low doses of antibiotics in a system, that tends to be where we have development of antibiotic-resistant organisms. Using antibiotics for the common cold, a viral infection, or other inappropriate conditions would be really probably one of the top reasons why we have the problem that we do, as well as the use of antibiotics in animal feed. The majority of antibiotics in the United States are used in animal feed. I think it's something like 70% of all antibiotics distributed in the United States are used in animal feed. Failing to complete the prescribed regimen, basically you're prescribed that dosage of the antibiotic for a certain period of time, and you need to keep the level of that antibiotic high in your system for that time to sustain that antibiotic and to completely kill all of the pathogen. Typically, this failure to complete the prescribed regimen goes hand in hand with using someone else's leftover prescription and then using outdated or weakened antibiotics. If you fail to complete your antibiotic, you might save that antibiotic and somebody else gets sick in your house, you can simply give that antibiotic to the other person. That should not be done because that could be weakened because the antibiotic is prescribed, the dosage is prescribed for that person's body weight. Another problem would be improper disposal, so flushing them down the toilet, getting into a city sewer system, which then goes to a wastewater treatment plant. So improper disposal would be another contributing factor. Now, what actually happens when the antibiotic resistant bacteria evolves, there can be basically 6 different mechanisms of resistance. So the bacteria might produce an enzyme that destroys or deactivates the drug. So your methicillin-resistant Staph aureus strains are usually doing this, producing an enzyme that destroys the methicillin. Slow or prevent the entry of the drug into the cell. So altered porin proteins in the gram-negative outer membrane can change the structure of that porin or the electrical charge associated with it to maybe repel the antibiotic. Alter the target of the drug so it binds less effectively, often seen with antimetabolics. So altering an enzyme active site, for example. Alter their metabolic chemistry, so abandon a metabolic step that's being targeted. So abandon the synthesis of folic acid, for example, and absorb it from the environment, which would be an example for that. So just abandon a biochemical process. Pump the antibiotic drug out of the cell before it can act. So these pumps can often pump out more than one type of antibiotic. Your multidrug resistant bacteria are usually capable of pumping out 10 or more different drugs. And then the last one would be biofilm production, which prevents the actual antibiotic from coming into contact with the bacteria. So a couple notes here. Antibiotics are effective against bacterial infections, and therefore they should not be used for treating the common cold or viral influenza. The antibiotic is completely ineffective. And this time of year, people are getting the sniffles and sneezes, and they go to the doctor requesting antibiotics. Depending on who your doctor is, a good doctor would probably send you home and tell you to take some cough medicine or something like that and treat the symptoms. So the antibiotic itself isn't going to affect the viral infection. It's going to be your immune system that kicks in to actually get rid of the virus. So here's a question: how are multi-drug resistant bacteria treated? So I mentioned that some antibiotic resistant bacteria can pump out 10 or more different antibiotics. So what if through multiple attempts there are no antibiotics that work? If you haven't already watched Hunting the Nightmare Bacteria, there are some examples in that video that show that there were no antibiotics that treated these people's infections. So what had to happen? One of the things that the doctors looked at were synergistic effects, so combining antibiotics to have a greater effect than either one of them alone. Amputation would be, removal of the infected tissue would be another strategy. New drugs, potentially. However, our antibiotic drug system is we're not creating enough new drugs fast enough to combat the problem of antibiotic resistant organisms. So the effective combination of drugs, synergism, like I said, that would be when the two antibiotics work better together than either of them alone. This is an example of a synergistic effect. There's 2 antibiotic discs here on the plate. The antibiotics seep into the plate and we create a zone of inhibition around the antibiotic. Here in the middle, where the 2 drugs actually interact and together, we have a much larger area that's created due to the activity of the 2 antibiotics together. This will conclude the Chapter 11 video. Make sure you complete the study guide and do the online quiz associated with this chapter.